We will now switch the field a little bit to MRI histology. Our last speaker is Christiana Tisca from the Wellcome Center at the University of Oxford. She does neuroimaging in mouse models and combines MRI with histology. She shares all the tools and protocols in her studies openly. And in her talk, she will introduce us to the current techniques employed in MRI histology preclinical studies. So we are looking forward to your talk, Christiana.
Thank you so much. I'm very excited to be here today. So yeah, we're going to switch gears a bit. In this presentation, I'm going to try to convince you why it's worthwhile to conduct MRI histology studies, and particularly in rodents. We'll kick things off with an introduction to the basics of histology. We'll then proceed to discuss the value that histology can bring to MRI datasets, followed by a description of some of the main challenges faced by MRI histology studies and solutions that our lab is proposing. I will then show you some example MRI histology work from our lab. And finally, we'll wrap things up with a short discussion on some important considerations for the future of MRI histology studies. And in line with the previous talks that we've had so far, I'm also going to be focusing on neuroimaging, but a lot of the principles that I will be discussing are directly applicable to other disciplines as well. So what is histology? Histology is an area of microscopy that studies the microanatomy and microstructure of tissue. In histology, we use different stains to generate image contrast, and these stains can be obtained in various different ways, such as, for example, immunostaining. So here we have proteins. Proteins in tissue that are labeled using specially designed primary antibodies that have an affinity for a particular part of the proteins of interest. So for instance, in this stain, the antibody is targeting some myelin proteins, and in this particular stain, it's targeting some polymer proteins that are found within neurofilaments that are found in neurites that are part of neurons. But you can see here in this case that we have that the stain has the same color, and this is because the same chromogen is used that is attached to the secondary antibody that ends up being attached to the primary antibody. So it's a bit complicated, but a similar principle. Sometimes, however, people are interested in using a different stain. So I'm showing here the Luxol Fast Blue stain, which is a chemical stain that targets myelin as well. So it targets the myelin sheath. It generates a similar contrast to this particular stain, but is obtained in a slightly different way. There is an extremely long list of stains that we can get and what they target. And final example that I'm going to show to contrast with what I've shown so far is the Nissl stain, which targets the DNA and RNA in cell bodies. So histology workflows, histology protocols have very elaborate workflows. Here I'm going to show the paraffin embedding protocol, and this starts with removing the tissue of interest from, let's say, the skull, if we're interested in the brain, so dissection. This is followed by brain cutting, so sometimes we're only interested in one section of the brain. And then this process is followed by processing and embedding, which is a protocol where the brain is taken from its fixation state through to various alcohol and other intermediary solutions, and finally put into wax, where it is embedded. And we then slice the brains and stain them using, in this particular case, immunostaining. And as I'm emphasizing here, the different components of the histology workflow can impact relating this data to MRI in different ways. For registration, for example, it is essential to maintain the brain morphology, and for stain quantification, we need reliable and consistent staining. And while in this talk, I'm going to keep focusing on paraffin embedding and immunostaining, some of these workflow steps are found in other methods, such as immunofluorescence. So in the context of MRI histology studies, we can now start to see that there are some necessary components. The first aspect that we need to consider is having tissue preparations that are compatible for both modalities. For example, mouse brains prepared for ex vivo MRI are usually perfusion fixed and doped with a contrast agent. And we need to ensure that the subsequent histology processing works well, considering the quality of the MRI tissue. Secondly, we need large enough regions of interest in order to establish a correspondence between the two modalities. And finally, to quantitatively compare the two modalities, we need some form of quantitative histological metrics. But we can also want more in order to conduct more elaborate studies, such as substantial brain coverage, good gray-white contrast in the histology stains that we obtain, co-registered data sets, which will facilitate a voxel-wise mapping between the two modalities. And ideally, we would like to go from 2D slices, which we normally get, to 3D histological data sets, which can be registered to MRI. So why is histology valuable, and what can it bring to MRI studies? Well, firstly, while MRI is very sensitive to a broad range of tissue changes, it lacks specificity, and validation studies can enhance the value of MRI by helping identify the biological source of signal change. Ex vivo MRI is usually used as an intermediary between in vivo MRI and histology, as it offers us the same signals as in vivo MRI and has the same tissue state as our slices that we obtain in histology. Another advantage of histology is that we can have multiple stains. So for example, a change in one particular MRI parameter can be due to multiple changes in tissue microstructure that happen at the same time. And traditionally, MRI histology research has focused particularly on relating myelin histology to MRI, hypothesizing that that is a primary driver of MRI signal change. However, we now know that the whole picture is a bit more complex, and in order to understand the different biological contributors to MRI signal change, we can acquire these multiple stains from neighboring brain slices and relate the different features captured by the different states to the MRI signal change of interest. Histology also allows us to characterize tissue at a different scale, which can be used to inform MRI simulations and computational models. The resolution gap between the two modalities is quite impressive. So in rodent brains, for example, we can go from about 100 microns cube in MRI voxel size to about 0.25 microns squared in histology data. And you can see here how much more impressive the detail is in histology data compared to MRI. And a further point is that histology can help us bridge our scientific understanding of a particular tissue of interest across species. There are very, very few human MRI histology data sets available, but it's relatively much easier to obtain MRI histology data in animal models, in particular in rodents. In mouse models, for example, we can invasively manipulate tissue microstructure, such as, for instance, completely remove myelin, and ask very specific questions about how this alters the microstructure and the respective MRI signals. And this can help us better understand the observed MRI signal changes that we see in humans. We should note, though, that there are strengths and weaknesses in both modalities, and combining MRI and histology can help us leverage their complementary strengths. But we need to think very carefully about how we can achieve that. For example, while histology data gives us a large number of stains and contrast, it is predominantly limited to 2D and is invasive. MRI provides us a non-invasive imaging modality, three-dimensional insight that can be particularly insightful in some contexts and help us better understand our histology data, as we're going to see in a bit. So what does our ideal data set look like? In order to fully benefit from the advantages of both MRI and histology, we would ideally want to acquire multiple MRI modalities and multiple histology stains in the same brain. So diffusion-weighted MRI structural imaging, quantitative susceptibility mapping, parametric mapping, as well as the multiple histology stains that I've shown you earlier. We want artifact-free high-resolution MRI data, and as MRI scientists, we're quite familiar with that. This is not as tricky to achieve as having whole brain histological slices with minimal staining artifacts. And we want our data sets to be fully co-registered, and we want reliable quantitative histological staining metrics that we can relate to MRI metrics at the MRI voxel level. So what are some challenges that we face here, and some solutions that our lab has come up with? where firstly, one of the main challenge that we face is the fact that we have variable histological protocols, and the fact that histological methodology is very poorly described, particularly in MRI histology papers. And one particular issue that we faced is that we've had to adapt standard processing and embedding protocols to work well with samples that were prepared for ex vivo MRI. And because we realize that this is a challenge that multiple labs will face, we've recently published our optimized tissue processing and paraffin embedding protocol that works well for mouse brain samples prepared for ex vivo MRI. And here are a few things that we've had to tackle. So for instance, if we use standard histological protocol that is used for fresh brain samples and process our brains using that protocol, we see that the resulting stained brain slices have a lot of artifacts and severe changes in morphology. And this will particularly impact our registration to MRI. What we want and what we've achieved using our optimized protocol is this these stained slices where the morphology is maintained, where very few folds are present, where the brain doesn't rip apart. Another goal that we've had was to maintain staining quality and uniformity. And what we found throughout our optimization experiments was that each individual stain had to be separately optimized, and standard protocols do not work well with the tissue that we work with that was initially prepared to be scanned for MRI. So as you can see here in this data here, the staining is quite faint, so there's a lot of non-specific staining compared to this. The contrast between white and gray matter is reduced, and you also have some other artifacts present. And with our optimized staining protocols, we have much more consistent staining, reliable, that we can trust when we relate it back into MRI data. And so once we acquire this data that has hopefully as few artifacts as possible, the following challenge that we need to address is extracting these quantitative staining metrics, which, as I've mentioned, are necessary for these MRI histology analysis. And one problem that this particular area faces is that subjective metrics are still common practice in some labs, including, let's say, cell counting by hand, which has been done up until a few years ago, or even expert operator thresholding for stain segmentation, which is still a standard process. But these practices are firstly not replicable across labs and are time-consuming. So in order to address this, our lab has developed an automated pipeline for obtaining a quantitative staining metric called stain area fraction. So for each individual stain, we get stain area fraction maps, and the stain area fraction represents the fraction of stained pixels within a given area. And these quantitative maps can be directly registered to MRI, which leads us to the next challenge that we face. And frequently in MRI histology analysis, ROIs are delineated manually, separately for each modality. And this is, again, an error-prone and time-consuming approach. And we therefore want automatic co-registration between MRI and histology, which further gives us the ability of resampling automatically derived ROI segmentations from MRI data into the histology native space. And for this in our lab, we use the Tensor Image Registration Library, or TERL, which now comes as part of FSL and enables automatic, straightforward registration of MRI histology datasets, including sparse single slice histology data. So even if you have one single slice for one brain, you can register it easily to MRI data. And TERL has so far been used in human postmortem and macaque MRI histology registration. And we've recently made some adaptations to TERL so that it works well with high resolution mouse imaging data as well. And we usually run MRI histology registration using a histological stain that has good gray-white matter contrast, which aids the registration algorithm. And once this problem is sorted, we can then run histology to histology registration. So as long as we have neighboring stain slides that are hopefully within the same MRI voxel, we can straightforwardly register the other stains to the myelin stain. So what are some applications? In discussing these, I'm going to be focusing on some work that we are conducting at WIN. And this work relies on having optimized protocols and automatic stain quantification and MRI histology registration. So MRI histology correlation at the MRI voxel level are one way in which we can start understanding the relationship between the quantitative metrics extracted from both modalities. And when we conduct such studies, we find that multiple stains, so here we have neurofilaments, myelin, PLP, and another stain that targets an extracellular matrix protein, we find that all of them correlate with, here in this case here, the same MRI metric, and we find that they correlate with multiple MRI metrics. This is very interesting in itself, but and encouraging, but we need to narrow down on the biological drivers of MRI signal change and disentangle the individual contributions of various different stains to MRI signal change. And one way in which we can estimate the unique variance of each MRI parameter explained by a given stain is to conduct partial correlation analysis. And another approach that we're currently working on is this mediation model, which we use when conducting MRI histology analysis in genetically modified rodent models. Our model allows us to separate the relative contributions to differences in MRI that are due to genetics and the different stains. So here I'm showing the myelin PLP stain and the neurofilament stain. And we want to be able to disentangle how these contributions compare. And a final example from our lab is some work that we're conducting using a very comprehensive MRI histology data set acquired in a single brain. That's called Big Mac. And in this specific study, Silay and Amy developed a method for hybrid diffusion-weighted MRI microscopy, where they combine information from both modalities to reconstruct 3D fiber orientations at much higher resolution than that offered solely by diffusion-weighted MRI. And they're using this approach in order to improve stratigraphy analysis. So finally, what are some things that I would like to see in the future? So firstly, I also believe in open sourcing tools, protocols, and data sets. This ensures replicability and fosters our scientific progress. And secondly, by building more and more comprehensive MRI histology data sets, we can start developing better histologically informed simulations and computational models of brain microstructure, which can benefit MRI. And finally, as we've seen briefly at the beginning, ideally we would want histology to match the coverage of MRI. And we can do that by acquiring 2D histology of the whole brain, which is time consuming, or by acquiring by using 3D microscopy methods such as Clarity. Both approaches come with significant challenges, so we need to still work on improving our workflow in order to facilitate this. And finally, everything that I've presented today, all the protocols and all the code is openly available, and all the data will be released. So we have this Win Digital Brain Bank platform where we're currently putting up all our data sets that you as well can access as long as you sign an MTA and are able to explore and play with and replicate our results if you're interested. Thank you. And if you have any questions, let me know. It doesn't have to be here. Please get in touch if you're interested by email as well. I'm very keen to discuss this topic. Thank you.